On the Yakult website, the question is, how does drinking Yakult help maintain my health? Of course, different categories. In category D, it enhances our body immunity to reduce risk of infections and reoccurrence of cancer cells. It looks like a health claim. If they are going to submit this as a health claim based on this evidence, it will be rejected. Because here it shows that taking Yakult for 3 weeks leads to an increase in natural killer cells. That's true. So it supports your natural immune system. But whether that reduces the risk of infections on one hand, or even the reoccurrence of cancer cells, you are not allowed to conclude that from this experiment. This also relates to the discussion we had this morning. Be careful in what you want to claim. This would be over the top. So what is the science? It's the same slide I showed this morning. What is the scientific literature, and specifically, what are the funding data in vitro, in animal models, and clinical data? And I'll only give you a couple of examples. Very nice data from the cover of the PNAS 2 years ago, showing that when you infect mice with Listeria monocytogenes, they grow in the peritoneum. When you pre-treat those rats with Lactobacillus salivarius, Listeria doesn't grow. This is a black and white difference. And why is this an interesting paper? Because we know why this is caused, because if you knock out the gene producing the bacteriocin, then it's ineffective. So here we have another example of a probiotic strain where we know exactly what the functional molecule is. And then cause-effect relationships will become easy to demonstrate. Another example from the work of Femke Lutgendorf, not published yet, also discussed this morning. What about gut barrier integrity? Here we have looked at expression of 2 tight junction proteins, occludin and claudin, which are normally very nicely expressed, of course, in the gut. This is in animals which we subject to a stressful situation called water stress, and then you see immediately loss of gut barrier integrity by reduced expression of tight junctions. When you pre-treat the animals with probiotics, in this case a mixture of several different strains, then you maintain gut barrier integrity, which is important for preventing infections that could occur during stressful situations. What about the immune system? What can you measure of biomarkers of the immune system? Again, looking at PubMed, what has been done. This is the top 5. Phagocytosis, secretory IgA, regulatory T cells, cytokine production panels, by far number 1, and response to vaccination. I'll show some data on regulatory T cells, cytokines, and vaccination. Immune system is a complicated system. I'm not going to explain that all to you. You must know that there are at least 3, even 4 different subsets of T cells. T helper 1 cells making a given set of cytokines, T helper 2 cells making other cytokines, and regulatory T cells in the middle who keep care of the balance. Example I want to give is an allergic disease. What you see in an allergic disease is there is too much production of T helper 2 cytokines. And the reason is that there is too little activity of regulatory T cells. So when we started out by selecting probiotic strains in order to prevent development of allergy, we were looking for strains that were able to downregulate T helper 2 cytokines, in this case IL-5. And that can be done by the fact that probiotic strains, selected ones, are able to induce regulatory T cells. And this is another example showing that not all probiotic strains are the same, they are all different. In this case, they differ in the capacity to induce regulatory T cells. And finally, in terms of biomarkers, Erik mentioned it already this morning, the response to vaccination. When probiotics would be able to increase the antibody response to vaccination, that would be a beneficial effect. And one of the few accepted correlates of protection. But here we have to be careful, in my view, because you have national vaccination campaigns for children. And those vaccination campaigns are designed so that everyone, we hope, will be protected in the end. And in order to interfere with probiotics would suggest that without probiotics, the vaccination campaign would not be sufficient. So we don't want that. But there's another kind of vaccination that's shown here, that's influenza vaccination. In the Netherlands, we only give influenza vaccination to risk groups, like the elderly. The elderly are at risk because their response, the open bars here, is very low. This is seroconversion rate. It's 2%, 10%, 5%. So that's not very good. And in this example, you can see that when you give probiotics to that population and then vaccinate, you see a significant improve of the vaccination response, which is a health benefit. In elderly, which are otherwise healthy, but have an increased risk of getting influenza infection. What are real clinical outcomes? That is a reduction of infection. And for instance, fever episodes here in infants, or other clinical outcomes in at-risk populations, reduction of eczema, landmark publication of Marco Kalliomäki in the Lancet. These are our own data from last year, Titia Niers published. In the field of prevention of allergic diseases, there is a rare publication, this one from Christine Wickens, where in one study 2 different probiotic strains are used with a clinical endpoint. Same design, same population, of course, because it's the same study. And there you see that in this case rhamnosus is effective, while Bifidobacterium lactis is without any effect. Again, reiterating, not all probiotics are the same.